Good morning, students. In the last class, we have discussed about the cell division. In that, we have discussed about the mitotic cell division. Mitotic cell division, as you know, that takes place in the vegetative cells or somatic cells. Now, see the second type of cell division which takes place in our body, that is meiosis or meiotic cell division. Basically, that takes place in reproductive cells. It is confined to particular cell and takes place at a particular time. Cells of sexually reproducing organism undergo meiosis. It produces gametes in animals and spores in higher plants. The cells in which meiosis takes place are called meiocytes. Example: oocytes, spermatocytes, sporocytes. Meiosis consists of two divisions that takes place in the rapid succession, with the chromosome replicating only once. One parent cell produces 4 daughter cells, each having half number of chromosomes and half amount of nuclear DNA. It is known as reduction division. Two divisions are known as meiosis 1 and meiosis 2. Meiosis 1. It has 4 phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1. It is more complex than mitotic prophase because of recombination that occurs in it. It may extend over weeks, months or years. It has 5 sub-stages: liptotin, zygotin, pakitin, diplotin and dikinesis. Liptotin. Chromosomes appear as thin thread by condensation. Each chromosome is double, consisting of chromatids due to DNA replication during interphase. Chromatids are closely adhered together. Zygotin. Homologous chromosomes come to lie side by side in pairs. Pairing is called synapsis. A regular space of 0.15 to 0.2 micrometer wide exists between synapsed homologous chromosomes. The space have highly specialized fibrillar organelle that is known as symptonemal complex. Pakitin. Chromatids of synapsed chromosomes slightly separate and become visible. Chromosome with 2 chromatids are called dyad. Group of 4 homologous chromatids called tetrads. 2 chromatids of same chromosome is called sister chromatids. Those of 2 homologous chromosomes is called non-sister chromatids. Crossing over occurs in this stage. It involves mutual exchange of corresponding segments of non-sister chromatids of homologous chromosomes. Diplotin. Homologous chromosomes separate at different places. This is called disjunction. Chromosomes do not separate at certain points called chiasmata. Dikinesis. Chromosomes condense again into short, thick rodlets. Centrioles move apart in pairs to the opposite ends of the cell. Asters form around each centriole. Spindle develop between the centrioles. Nucleolus disintegrate. Nuclear envelope breaks down. Tetrads are released in the cytoplasm. Now see the metaphase 1. Tetrads scattered in the cytoplasm move to the equator of spindle. They align in 2 parallel metaphase plates. Each homologous chromosome has 2 kinetochores. Both are connected to same spindle pole. 2 kinetochores of homologous chromosomes act as a functional unit of metaphase 1. Anaphase 1. The homologous chromosome separates while sister chromatids remain associated at their centromeres. Telophase 1. The nuclear membrane and the nucleolus reappear. Cytokinesis follows, and this is called as dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic division is called interkinesis and is generally short-lived. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. See the division of cell. Meiosis 2. Prophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles as normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2. At this stage, the chromosomes align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatids. Anaphase 2. It begins with simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move towards opposite poles of the cell. Telophase 2. Meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells. See the formation of 4 haploid cells. Haploid cells means the chromosome number becomes half. Significance of meiosis. Meiosis is the mechanism by which conservation of a specific chromosome number of each species is achieved across generation in sexually reproducing organism, even though the process per cell paradoxically results in reduction of chromosome number by half. It also increases the genetic variability in the population of organism from one generation to the next. Variations are very important for the process of evolution. That's all.